Understanding organs on a chip, a miniature revolution. Imagine shrinking a human organ to the size of a computer chip. That's the essence of organs on a chip. These tiny devices are revolutionizing medical research. They're not actual organs, but rather sophisticated models. These models mimic the structure and function of human organs. They're designed to help us understand how organs work and how they react to various stimuli. Organs on a chip are typically made of clear plastic. They contain tiny channels lined with living human cells. These channels are arranged to replicate the structure of organs. For instance, a lung on a chip might have two channels. One represents the air sac and the other represents a blood vessel. This setup allows researchers to observe how the organ functions in real time. The beauty of these devices lies in their simplicity and effectiveness. They provide a controlled environment for studying organ function.
The journey from Petri dish to chip, a brief history. The concept of organs on a chip didn't appear overnight. It evolved from decades of research in cell biology and microfabrication. In the early 20th century, scientists began growing cells in Petri dishes. This was a significant step forward in biological research. However, these flat cultures couldn't replicate the complex three-dimensional structure of organs. As technology advanced, researchers sought ways to create more realistic models. In the 1990s, the field of tissue engineering emerged. Scientists began growing cells on three-dimensional scaffolds. This approach allowed for more complex tissue structures, but it still couldn't capture the dynamic nature of living organs. The turning point came with the development of microfluidic technologies in the early 2000s. Microfluidics allowed scientists to control and manipulate tiny amounts of fluids. This technology opened up new possibilities for creating organ models. Since then, the field has exploded with innovation.
Why chips trump traditional models. Traditional research models have long been the backbone of medical science. These include in vitro cell cultures and animal testing. While these methods have yielded valuable insights, they have significant limitations. In vitro cultures are too simplistic. They can't replicate the complex interactions within an organ. Animal models, on the other hand, don't always translate well to humans. What works in a mouse might not work in a person. Organs on a chip offer several advantages over these traditional models. First, they provide a more accurate representation of human physiology. The chips use human cells and mimic the structure of human organs. This makes them more relevant for studying human diseases and treatments. Second, they allow for real-time observation and measurement. Researchers can monitor changes in the organ-like tissues as they happen. This approach offers a level of precision that was previously impossible with traditional research methods.
Microfluidics, the heartbeat of organ chips. At the core of organs on a chip technology lies microfluidics. This field deals with the behavior of fluids at the microscale. In simple terms, it's about controlling and manipulating tiny amounts of liquid. Microfluidics is what allows these chips to mimic the dynamic environment of living organs. It's the key to creating realistic organ models on such a small scale. Microfluidic devices use a network of tiny channels and chambers. These channels are typically smaller than the width of a human hair. They allow for precise control over fluid flow. This is crucial for replicating the conditions inside organs. For example, in a liver on a chip, microfluidics can simulate blood flow through liver tissue. This flow delivers nutrients and removes waste products, just like in a real liver. The principles of microfluidics are based on fluid dynamics at small scales. At this level, fluids behave differently than they do in larger volumes. Surface tension and capillary forces become dominant.
Section 5. A tour of the body on a chip. The world of organs on a chip is diverse and fascinating. Researchers have developed chips for various organs and tissues. Each chip is designed to mimic specific aspects of organ function. Let's take a tour of some of the most interesting examples. Think of it as a miniature journey through the human body. Let's start with the lung on a chip. Imagine a sandwich made of clear plastic. The filling is a thin, flexible membrane coated with lung cells. On one side of the membrane is air, on the other, a blood-like fluid. The chip can stretch and relax, mimicking breathing. It's like a tiny, see-through lung in a box. This chip has been used to study respiratory diseases and test new drugs. Next, we have the heart on a chip. Picture a tiny pump that beats like a real heart. It's made of heart muscle cells that contract rhythmically. The chip can measure the strength of these contractions.
Section 6. Mimicking Nature. The Art of Biomimicry. Biomimicry is at the heart of organs-on-a-chip technology. It's the practice of emulating nature's designs and processes to solve human problems. In the context of organ chips, biomimicry means creating artificial environments that closely resemble those in the human body. This approach allows researchers to recreate the complex interactions that occur within organs. The goal of biomimicry in organs-on-a-chip is to replicate the microenvironment of human tissues. This includes not just the physical structure, but also the chemical and mechanical cues cells receive. For instance, in a bone on a chip, researchers might apply mechanical stress to mimic the forces bones experience during movement. In a brain on a chip, they might create specific chemical gradients to guide neuron growth. One of the key aspects of biomimicry in these devices is recreating the extracellular matrix, or ECM. The ECM is the support structure around cells in the body. It provides physical support and biochemical cues. In organ chips, researchers use various materials to mimic the ECM. These might include hydrogels or specially designed polymers.
Section 7 Technological Marvels 3. Bioprinting and Smart Sensors. The field of organs on a chip is constantly evolving, driven by technological advancements. Two areas that have significantly impacted the field are 3D bioprinting and smart sensor integration. These technologies are pushing the boundaries of what's possible with organ chips. They're allowing for more complex and accurate organ models. 3D bioprinting is revolutionizing how we create organs on a chip. This technology allows for precise deposition of cells and materials in 3 dimensions. It's like building a tiny organ layer by layer. With 3D bioprinting, researchers can create more complex tissue structures. They can precisely control the placement of different cell types. This leads to more realistic organ models. For example, 3D bioprinting can create blood vessel networks within organ chips. This improves nutrient delivery and waste removal, just like in real organs. Smart sensors are another game-changing technology in this field. These tiny devices can be integrated directly into organ chips. They allow for real-time monitoring of various parameters.
Section 8. Reinventing drug discovery with organ chips. Drug discovery is a complex, time-consuming, and expensive process. Traditional methods rely heavily on animal testing and simple cell cultures. These approaches often fail to predict how drugs will behave in humans. This leads to high failure rates in clinical trials. Organs on a chip are changing this landscape. They're providing a more accurate and efficient way to test new drugs. One of the key advantages of organ chips in drug discovery is their human relevance. These chips use human cells and mimic human physiology. This makes them more predictive of human responses than animal models. Researchers can test drugs on lung-on-a-chip devices, for instance, to see how they affect lung function. They can observe in real time how the drug is absorbed, distributed, and metabolized. Organ chips also allow for more nuanced drug testing. Researchers can study not just the main effects of a drug, but also its side effects. For example, a heart on a chip can be used to test for cardiotoxicity, a common side effect of many drugs.
Section 9. Modeling diseases, a microscopic view. One of the most powerful applications of organs on a chip is in disease modeling. These devices allow researchers to recreate disease conditions in a controlled environment. This provides unprecedented insights into disease mechanisms and progression. It's like having a window into the human body at the cellular level. Disease modeling with organ chips starts with the basics of the disease. Researchers introduce disease-causing factors into the chip. This might be genetic mutations, pathogens, or environmental factors. For example, to study asthma, they might introduce inflammatory molecules to a lung on a chip. They can then observe how the lung tissue responds. This approach allows for detailed study of disease onset and progression. One of the key advantages of this approach is the ability to study human diseases that are difficult to model in animals. Many human diseases don't have good animal models. Organ chips provide a human-relevant alternative. For instance, researchers have used brain-on-a-chip devices to study Alzheimer's disease. Section 10. Unraveling biological mysteries. Organs on a chip are not just tools for drug testing and disease modeling. They're also powerful platforms for basic biological research. These devices allow scientists to study fundamental biological processes in unprecedented detail. They're helping to unravel some of the most complex mysteries of human biology. One area where organ chips are making significant contributions is developmental biology.